Why does it happen if you have so you have the competing hydrogen bonds in solvents? And how is it that you still see almost always secondary structure in a...
Are you asking how or why?
Why, right.
Because that way they can bond better to other structures or something?
No, it's much more of a physical reason. So it's really physical forces if you think about it.
Lower energy state?
It's a lower energy state, yes and no. So you need to consider that the energy difference between the hydrogen and the solvents and the hydrogen bonds backbone isn't very large. So the difference in energy is very small between solvents, hydrogen bonds to the backbone, and the backbone hydrogen bonds. So it's a small part of the reason, but that's not the reason why you see 80% of the amino acids typically in a secondary structure type. 70 to 80%. No, also not quite.
Maybe that's when like these reagents are formed, some they probably pack some hydrophobicity away, or
yeah, you're getting closer. So if we consider this figure maybe. So what happens if we keep this, say we went straight, we just have one straight lying backbone chain here right in the middle, so it's not in the structure, so we have a stretched one going right through the core of this protein. Say it did have a large hydrophobic groups, so it's quite hydrophobic, but it just has its backbone stretched out through the middle of this. Is it going to be stable, or is there a problem there?
It's not going to be stable.
Why not?
Because the water doesn't want to interact with the hydrophobic residues. So basically, but it has to sit against the hydrophobic residues. So basically, it's limited in its movement because of it, and it's not.
Yeah, nearly. So, but so if you have the stretched backbone, you still have these hydrogen bond donors and acceptors, right, in the middle of your hydrophobic core. So now you have hydrogen bonds or hydrogen donors and acceptors, so satisfied ones, that sit in a very hydrophobic environment. So that's not going to be favorable, right? So do you see the problem? So if you have the backbone and you have unsatisfied hydrogen bonds in the middle of your protein in your hydrophobic core, there's no donors and acceptors there. There's no water, so you don't have this competing. There's just nothing. They're actually broken, and then it becomes energetically very unfavorable. So what happens instead is that, as you see here, you get these helices that make sure that the hydrogen bonds in the backbone are satisfied, and then the hydrophobic residues can start sticking together. Because actually, because you have this hydrophobic core, they need to be satisfied by the backbone because there's no water anymore. Yeah, that's the easier way of thinking about it. Alright, so we've seen this. So this is a quaternary structure. And so what you see here is, I like this figure because you can actually really see from the structure that this is a motor. It actually does really rotate in the cell. But what the point here is actually that you see there's different colors here, and every molecule, so every single molecule, so every separate protein here is a different color. So you see that it's actually a huge complex of different proteins binding specifically together. And also from the structure you can immediately see that this needs to be stuck together in this form with these interfaces between these proteins to make this a structural unit and a working complex. Yeah, so this is usually what we refer to as a quaternary structure. Okay. So then the classic picture, and we'll stay did that for a while, is that we have a structure-function relationship. So the idea is that the protein can only function, it's quite clear here, once it's correctly folded in its territory and maybe also its quaternary structure. Yeah? So it's taken on the right form, and then it can perform its function in the cell. Does anyone know any exceptions to this? So about 30% of the sequences in, or own proteome don't actually fold at all. But that's something we'll see in later courses. So, but there's hugely large unstructured regions there that you just look like, and they also sit around in. Okay, but for the majority of proteins, and specifically everything we'll look at in this course, so all the structures, these structures or these sequences, they fold, and they need actually to fold into a specific structure to perform their function. So for now, you can assume that this is the relationship you're interested in. And so you get this kind of thing. So you have the translation here into an amino acid sequence, you have the folding, and then it can function. Right, and we already said enzymes, quaternary structures, so you have different ways it can interact with its environment. This is just a picture of the complexity of the network. So if you, I'm sure you've seen this before, you see the in small here a protein-protein interaction. So here I have one protein interacting with the blue one, actually here 3, here 4 interacting together. And of course you can start drawing the proteins as just single dots, and then you get a huge network of proteins that sometimes in different times, in different places, can interact with each other. These huge connected networks. Yeah? So there's a lot of protein-protein interactions going on. And we can actually measure this also high throughput now. Okay, so I said I was going to come back to this. So can you think of any problems that we may want to solve computationally on protein structures? Yes?
Maybe what we've seen since we're now, if we start with an amino acid sequence, and for protein folding prediction.
Protein folding prediction. So we have a sequence. Actually, someone already mentioned it way earlier when we had the example of the PDB, and we want to start predicting what the tertiary structure is. Yes? And it's still an unsolved problem. So we'll see that in structural bioinformatics, but that's a problem scientifically we can't do yet. Neither with physics nor with machine learning. We're quite good at it when the biology helps us, but when we don't have good examples, so when we don't have a PDB structure there that looks a bit like your sequence, we're lost. So we can't really construct it from scratch. Yeah?
What is actually the problem? Because the physics is known. What is the problem?
Complexity of the physics. The complexity of the physics. So if we can't just we can't simulate it, for example, for a long enough time scale to be able to predict it.
Is that a computation problem then?
Yeah, both a computational and a parameterization problem. But it will. We're going to go into this in structural bioinformatics for a long time, so I can talk about for 4 weeks about this. So let's not do that right now. But it's still a large problem, and there's many facets to it. There's also parts that we set new steps, and a couple of really nice recent steps have been set to get better at this, but it's still a very difficult problem. Was another question? Okay. Any other ideas? Yeah?
Protein dynamics.
Protein dynamics, so yes, so that's molecular simulations, molecular dynamic simulations, yes. Anything else? Maybe even, yeah?
Interaction site predictions, maybe?
Yes, so exactly. So can we predict this or this? Exactly. Also very difficult. Can't do this yet from sequence at all. Structure also very difficult. It's called docking. Works a bit. Anyone else? It's actually a simple problem that's more similar to alignment. Can you imagine? Homology modeling, that's also structure prediction. And that's based on alignment, yes. So we also have something called structure alignment. So if we want to compare 2 protein structures, so for example the shape of the red one and the blue one, we actually want to structurally align them just based on their topology. And see, that's computationally a very difficult problem. It's much more difficult than sequence alignment. Which is also again something we'll look at in structural bioinformatics. There's one obvious one I haven't heard yet. It's also prediction. So exactly, prediction of the secondary structure. This is a much better or a much easier problem, and we can do that up to about 80% accuracy at the moment. So that's something that I would say, well, it's almost as good as you may be able to get theoretically with also the uncertainty and annotations. So that we can do quite well. Okay. I think we've had most of them. Okay, so this is the first one. Given a sequence predictable the structure for protein, you can think about the tertiary structure or the secondary structure. And then the other problem I just said is what if we want to compare two structures and we want to see how similar they are. Now this problem is really important also for the lecture today and for your course at the moment in your project, because it's actually this type of algorithms, and we just now consider the output, and we basically consider the annotated and curated output. So this is what FoB uses either manually or sometimes they do use computationally to see if these structures align, and then they look at these structures to see if two structures are similar. So this is the kind of way you can see if a structure A and a structure B are similar. You really try to rearrange them in such a way that they fit on top of each other. Yeah? So like here, so these are two different structures. You see they're alike, but if you look, for example, carefully at the loops here, you see that these loops here are longer than these loops here, and they are different, so they're not quite the same. Then if you align them, you see that especially the secondary structured parts align really well, but you also see that, for example, the blue loops here extend higher and also here lower than in the magenta one. You see that? Now, this is a very specific alignment. These are actually two different proteins. We have a rhodopsin 1 here and a second rhodopsin in a completely different species. And these are actually having the same function. These are actually evolutionary related proteins. Now, if we have aligned them purely on the structure, so we just match the structure, we also get a sequence alignment out of this. This is the sequence alignment. So this is now not made with BLAST like you've seen before. You couldn't actually make this with BLAST. But it's made with a structural alignment program. And already when you look at it, you see that only very, very occasionally some letters overlap. There's a lot of gaps, and it's very, very difficult to spot any similarity between these two sequences. If I would have given you these two sequences, I don't think any of you would have been able to align these sequences in this way to each other without knowing that structures, or without knowing the structures. If we look here, you see that the sequence identity is 7.4%. That's very, very low. That's lower than random. So if we have two random sequences and we align them, then we typically get more in the order of 10 to 12% of sequence random. So this BLAST would never be able to align it. You give these two sequences, BLAST will not find this alignment. Yeah? So this is a very good way, so you've heard it maybe in words before, but this is a really nice example of where you see that structure is much more conserved than sequence. Yeah, so you see that the structure, it's clear, right? You look at it, they're clearly preserved, and we also know, so you can say, well, maybe some people may say convergent evolution, but the functions are also really the same of these proteins, so these are really evolutionary related. There's a lot of neighbors around it, so you can see the evolvement steps back. These are evolutionary related, you see it clearly in the structure, and sequence, there's hardly any signal left. Yeah, so you can hardly match them. Yeah.
Which percentage would be high or medium?
Typical cutoffs are around 30%. So below 30% becomes difficult.
Okay.
And above 30% it's quite easy. So then you get into the domain of BLAST again, above 30-40%. Yeah. This is also the reason why, on the other hand, very sensitive sequence alignment programs are in so much demand, because on one hand you saw there's only 70,000 PDB structures. We don't have many annotations of well annotated proteins where we actually know where the functional sites are. And so we have way more protein sequences, but we actually see that it's actually difficult to match the sequences to the structures. Because of course if we do it, if we could find this, and if we have something that is maybe more like 50% sequence similarity or 60%, then actually really be quite sure, so we can test around that these structures are the same. Yeah, so if you have two very similar sequences, typically the structure is the same. Because sometimes you have small loops that change, so sometimes even a small domain that's a bit different. And typically you will see very similar structures if the sequence is conserved. Yeah, so that once we have found a blast herbicide blast hit, we know that it will typically have a similar structure. Maybe one last point. So I showed the gaps earlier. Here you can actually also see where the gaps come from. Now it's maybe a little bit difficult to match, but can anyone guess where would the gaps in the structure line up to? It's the loops, right? So that's where you can of course more easily insert structure or a new sequence into the structure. So you almost always see that where you used to get large gap regions, it's on loop regions in the structure. This is also again why HMMs are so much more sensitive, because there's a very strong signal coming from the insertions and deletions in these sequences. Okay, so here this is a very good significant score for the structure alignment with very, very low sequence identity. So this is maybe one of the most important figures for this lecture, and it's a tricky one. So here we have taken a whole set of three-dimensional structures, and we've structurally aligned them. And here we have real homologous 3D pairs. And here we actually do sequence alignment for the red ones on just random sequences. So this is the distribution of sequence scores, pairwise sequence similarity in this case, or not identity but similarity, so this would be Gaussian or something, or scored by Gaussian. And then here you have the real distribution. Now you can also immediately see why it is so difficult to find hits with blasts. Because you actually see that for a very large set, so the kind of lower set here of the homologous 3D pairs, the random distribution completely overlaps. Yeah? So if you fall in this part here of sequence similarity, you will never be able to separate between a random chance, two random sequences, and a real 3D structure-based homology. Yeah? So based on sequence similarity alone, it's very difficult to decide whether something is a random hit or a real homologous 3D pair. Any idea what this blue line, in which database do you think this blue line has been made? What would you take to generate that one? It's one of yours. So it's either PFAM, SCOP, or Go. Which one would you use? SCOP. And so then we have the different levels in SCOP, right? Fold, superfamily, family, class, above class, folds, superfamily, family. Which level would you go for?
Probably family. Because in family you can really have proteins which have domains which have the same fold.
It's probably superfamily, because there you know the structure is the same and also there is evidence for homology. So that's exactly the criteria here.
What's the difference between family and superfamily?
The family in SCOP has a sequence cutoff. If I'm correct, I do it by heart now because I don't have slides. I think it's 30%. Could be 50. I'm not sure. So you need to. There's a good definition on it on the SCOP original SCOP paper, but there's a hard sequence cutoff for inclusion into family, and then superfamily is basically annotated. So they will only put families together in the same family if they say there's evidence in structure, for example, because or in function, that these families are evolutionary related. So they'll typically look at the conservation patterns in the functional sites to see if they are remaining in the same place. Okay. So, and the most important conclusion is actually it's really hard to distinguish just based on sequence similarity homolog pairs from random pairs. Yeah? And of course this has a direct influence on how, to what extent we can actually use BLAST to find these true homologous pairs. And so this is also why something like PsyBLAST, where you have these sequence profiles, just allowing you to probe a little bit further into this direction where your sequence similarity diminishes, and we can still get a signal here from the conserved residues. And so it's the same idea here. And you see again that HMMs go even more distance. Quick picture then here of HMMs, and you see here, you don't have to know this, but what you see is to model a sequence as we model the entire sequence in a HMM, and you have specific insertion and deletion states, so you can actually assign different gap penalties effectively, so it's insertion and deletion probabilities at specific positions, and that's what makes the HMM so much better again than the PsyBLAST. And of course, underlying all these kind of sequence-based methods, we should not forget that the vast majority of the signal, like the gaps and the conserved position, is sometimes coming from the function, but in many cases directly coming from the structure in terms of conservation. Okay. That should not be the end, because I had
see if it's back on there.
Okay, via the PDF then. So why do we care about protein alignments? So what kind of maybe more practical tasks? So now we've kind of looked at quite fundamental things in proteins, evolution, structure. What kind of more practical tasks maybe actually want to solve with alignments? So I'm actually thinking here about the biomedical domain. Can anyone think of? Sorry?
Find substitutes for molecules that we need.
Find substitutes, so maybe with homologous relationships we can maybe have proteins that have a similar function. I think that's still quite far away, but maybe with insulin, yes. I think so. It's like with actually the evolutionary conservation is a very important signal. And one of the ideas here is just that you can actually use, so this is a paper we looked at a while ago, and won't go into detail. It's just an example of how it can be used. And so you can imagine that if something is very conserved over evolution, and now for example you get a mutation in a cancer cell, that if that mutation hits a spot that's very conserved in an evolutionary profile, the chance that that does severe damage, or in other words, that severely disrupts the cell biology, is much higher than if it's in a very unconserved position. And these type of conservation scores are strongly used. So I think you've seen this use case before, right? You've discussed it, I think, in the data lecture. But it's actually for this use case that both profiles, evolutionary profiles, both at DNA level and at protein level, but even structure can be used to make inferences. Now, so what's the problem? So typically we take a tumor sample, we sequence that, actually that's going to be one of the next lectures, so we'll see how we generate, if you say, loads of reads, and then we see this tumor is different from the reference genome, let's assume for now from the patient's own genome, so we find a different position. Now the problem is that for a single tumor, we typically would find thousands of mutations. And they're not all driving the tumor biology. Why is that? So why do you find so many mutations in tumors?
Because they divide so rapidly.
Sorry?
Because they divide so rapidly.
Yes, and typically also the check system, so for not has gone broken. So actually they accumulate, so it's actually part of the tumor mechanism that they become chromosomally unstable. So they actually acquire mutations more than a normal cell would at every replication. So typical tumor cells would have thousands and thousands of mutations in there. So you find a lot. But what you now want to do is actually you want to find the ones that are really important for what type of cancer it is, to specify which mutations are actually really essential for the tumor biology. And so how are you going to find that back? So you do this, and then we find thousands of different potential interests, but we don't know which one we can maybe make treatment decisions on, and which are just completely randomly because they're hypermutators anyway. So it's the typical task you need to do here. So you need to find which positions are actually very likely to drive the tumor biology. One of the things to do is just get a lot of patients. So if you get a lot of patients, what you'll see is that at the same positions, that's why this bar is higher here, you see that for many patients at exactly the same spot, typically also in the same gene, you see mutations. And then apparently, okay, this part is enriched, so what you actually see is positive selection for a specific mutation driving the cancer. So that's how we get to functional genes in cancer. So we get these called driver mutations, and we can pinpoint where they are by comparing a lot of mutations from different patients. But now what's one of the problems is that there's many other genes that are also affected and also have been shown to actually driving, but they are much rarer. So if we have 50% of the patients affected by that mutation, it's easy to spot. We have a large tail of genes where only 5% of the patients have it. Now, are these random or are they actually important? Now, this is explaining driver mutations. Skip that for now, I already explained it. But what you can do now is actually you can align different genes. So you can look at the sequence alignment. And what you actually see is that the mutations not only on a single gene start being at exactly the same position in different patients, but also in homologous genes. So if you align homologous genes of known driver targets, you see actually at exactly the same spots in different patients, but typically at a lower frequency. You see also mutations accumulating. So this is actually a very nice way to look for the more rare type of mutations and see which ones are still very important and stood out and figured out by homology. Just as we looked at homology profiles before. And so this is what you see here. You see different genes here, and you see that at all these different genes here, exactly at this position, this is a large sequence family, here aligned, you only see the positions, but all these proteins are there. You see here that the substitutions here indicate that here are exactly fall at the same position in the alignment on many of the homologs of the known cancer genes. And so, okay, RAS is the famous one, and HRAS is a homolog, but these are also homologs of that protein that can actually drive the cancer. Okay, so that's also the end of the lecture. What do you need to know? So protein structures, primary structure, secondary tertiary structure. Think about bioinformatics can be used. Structure-function relationship. Also bioinformatics on there actually. Also do structure-function prediction. Structure more conserved for sequence. That one, of course, you need to really know. That's very central in the entire course. Cyblast, sequence profiles, and the idea of impact prediction.